But with that, we're very excited to move into section 3 of today's workshop, and I'd like to introduce Dr. Alba Grifoni to present using the IEDB in your research. Thank you very much, Alba.
Thank you, Nina, and hello, IDB users. I have been a very excited IDB user for over 10 years, and so the goal for me today is to give excitement about using IDB. And so I'm going to show you how different IDB features, such as the database or even prediction tools, can be used to help in your research. And so in the past years, I've been using different viral examples. This year, I would like to go back on SARS-CoV-2 a little bit with the provocative title that the pandemic isn't over. And so one thing that we haven't realized is that the SARS-CoV-2 has still a couple of things up their sleeves, and they keep mutating and evolving. And so there is this specific sublineage of Omicron BA.2.86 that has been classified as variant under monitoring, and this hasn't happened in a while. And part of the reason why that was decided is because of the amount of mutation in the spike protein that you have in these novel variants as compared with the BA.2 that originated from. And so have in mind these amount of changes are very similar to what we saw at the beginning between the original SARS-CoV-2 and the Omicron variant. And if we wanted to go over from the BA.2.86 to actually the original ancestral, we're talking about 60 amino acid mutation for the spike protein, and even more if you consider the entire proteome. So in a way, this suggests that it is important to define pipelines where we are able to predict what is going to be the effect of upcoming variants on our immunity. And so beautiful work has been done on the B cell side of things, and I'm a T cells aficionado, so I'm going to try to answer this question in the slide I'm going to show you today on the T cell side of things. So how can I answer this question using IDB? And so you can do it in two different ways. The first one is to essentially leverage all the fantastic data that over 100 different labs worldwide, and at this point probably even more, have been put in trying to define experimentally what are the SARS-CoV-2 epitopes. And the second part, and the second way to answer this question, is gonna try to predict the effect. And so I'm gonna go into that later. So for the first question, as usual, and you have been more and more familiar with the way we use IDBs, you are able to extract the SARS-CoV-2 epitopes by using queries on organisms, SARS-CoV-2. We are interested particularly in the human population, so that's the host that we select in this type of query. And then whether we are interested in CD4 or CD8, we can classify the restriction on class 2 and class 1. So this is the first step that is pretty much common in all our analyses. An additional step that we have implemented is to filter for canonical sizes, and that goes back on what Alex has shown you at the beginning of this workshop. It is important to go and define really what are the minimal epitopes, and that is particularly relevant in the context of SARS-CoV-2, considering the amount of data that has been generated. Finally, and this is another step that we have implemented, IDB keeps evolving with the science. And so 2 years ago, the majority of the IDB epitopes available for SARS-CoV-2 were directed against ancestral. But now more and more scientists have been starting to study also variant evolution. And so if we really wanted to have a clean comparison between what is the effect if we compare the ancestral original epitopes in BA.2.86, we have to make sure to do an additional filtering. And that is possible thanks to ImmunomeBrowser. And ImmunomeBrowser, as I'm going to show you later, allowed you to really visualize the mass of data that IDB has included as a function of the protein, but it also provides information about sequence identities. So how similar is the single epitopes with the reference sequence that is provided by the IDB, that in this case is spike ancestral. And so adding this third step allowed us to go to a fair defined list of SARS-CoV-2 epitopes, and then we start asking how many of these epitopes are affected by BA.2.86 mutation. And so these are the results that we perform. The first question that we ask is, what is the fraction if we consider all the protein of SARS-CoV-2. And so that is relevant because, as we know, T cells can recognize virtually all the different protein of SARS-CoV-2, particularly if the people experience infection. And so if we do this type of analysis, as you can see, 72% of our IDB epitopes are actually 100% conserved. So no problem at all, the mutation don't affect them even remotely. Of the 28%, we then ask, okay, how many of these epitopes have mutation? Because the number of mutation potentially can impact the capability, more or less, of the HLA to be able to still present the epitope to the immune system. And so as you can see, the majority in our case were 1 to 2 mutation. So that brings a little bit a different question of how much are these mutations impacting the of the HLA to bind the mutated epitopes. And so to do that, we follow a different type of query. The first 2 steps was the same, but in this case, we went to the assay tab to extract what are the HLA restriction of the epitopes that we define. And then we leverage a different component of the IDB, the analysis tool that Alex is going to talk to you about after me. And what we did essentially was to try to run prediction and see how the original epitope versus the BA286 epitopes changed looking at the same HLA molecules. And so this is the results of this analysis. On the x-axis, you have the prediction value for ancestral, and on the y-axis, you have BA286. We have previously shown that a fluctuation of 3-fold change is actually still tolerated. And so as you can see, 96% of these mutated epitopes are still retain the capability to bind to the HLA molecules, and only at 3% of the epitopes show an actual decrease. And so this is great, but we're looking at the entire proteome. I start my talk essentially telling you that the majority of the mutation are on the spikes. So what happened if we narrow down our query just to spike epitopes? And so here's are the results up for the different proteins, and here below you see the spike. So you do see that indeed in terms of 100% conservation, the number goes down. You have roughly 56%, half of it still 100% conserved. But once again, when you dive into the mutated epitopes, the majority have a couple of mutations per epitope, which when we look at the capability of the HLA to still be able to bind these mutated epitopes, we do still see again that the majority retain binding capabilities. What about CD8? So CD8 show both for the entire proteome and the spike specific a higher fraction of epitopes that are still conserved. The number of mutation per epitope is very similar to what we see on the CD4. However, in the context of CD8, as many of you might know already, even a single amino acid mutation can be sufficient to disrupt the capability of HLA class I molecules to still bind the epitopes. And so once we look at these data in terms of prediction, we see that still a larger fraction, 70% and 62% of the restriction is still retained, but the amount of decrease binding observed with mutated epitopes goes higher to 18% than 24%. So can we put this in context? And so to compare apple to apple, we went back to a study that we performed actually at the end of last year, where we were asking this question just looking at Omicron. And at that time, it was just out, it was the BA1 sublineage. We didn't know that yet. We were hoping Omicron was it, but it wasn't. And so what you can see is that a little bit what changed in terms of preservation between the original BA1 and the BA286 is that we start seeing a slight moderate decline on the amount of conserved epitopes. In terms of restriction, though, the message overall is the same, which is in general, there is retained capability of the HLA to still bind these epitopes. And so I'm gonna stop sharing my presentation and show you how can we extract the data ourselves on IDB. And so here is our beloved database. And so how I'm gonna be able to extract my SARS-CoV-2 epitopes. you can select the SARS-CoV-2 organism. You want to go for T cells because that's what we're interested in right now, and you want to look at the human host, which is what I select for my specific study. And so while I let the database run, I actually already ran the query, and as you can see, we select in this specific case class 1 restriction. That was another parameter that you could select at the beginning, or you can always go back and select it over here in the MXC restriction tab. And so as you can see, if we look at the CD8 epitopes, we have over 2,292 epitopes. What happens if we apply the linear peptide restriction in size? So you can do this over here, and I'm going to select what is recommended by IEDB, and this is in this tab the results of it. You can see that out of the 2,000, almost 300 epitopes we were having before, if we apply the filtering now, we go down to 1,700. So this filter really makes a difference in terms of selecting the epitopes. So what about the sequence identity that I've been showing you before? You can get that information by looking into the antigen tab. Here you're going to have your epitopes that are divided depending on the protein of origin, and by clicking over in this icon, we're going to be able to see that in ImmunomeBrowser. What happens if we look in the ImmunomeBrowser? So ImmunomeBrowser is going to give you a general visualization of your epitopes as a function of the entire protein. The response frequency is essentially the number of positive versus the number of tested. And here, in order to also show you the amount of variability, IEDB is plotting you the interval, the 95 percentile lower and upper bound. So essentially your RF score is somewhere in between this position. This is in terms of the donor, and right down you can have essentially the same question in terms of assays. If you scroll a little bit down, you can visualize your peptide of preference in 3 dimensions. That's particularly relevant in the context of B cells. And then finally, over here, you have the table that recaps essentially the information of the different epitopes, and it is your identity scores that is going to be able to tell you whether the peptide is really originated for the ancestral sequence or for a different variant or something else. And so in this case, our question was, by looking at the data that we generated so far, can I pinpoint specific region of the protein that are characterized with decrease? And so we did that. And let me show you my screen real quick. Sorry, one too fast. Okay, so here are essentially what you have seen in the context of ImmunomeBrowser. This is just a different graphical visualization. I'm also including the different region of the spike just to have a perception of where are the decreased region located. And as you can see, very interesting, not unexpectedly, the majority of decreased region are located both for CD4 and CD8 in the RBD region. However, we have some regions that are located in the NTD domain. Nevertheless, the peaks of response that you observe essentially tell you how frequently that particular region is recognized in the population. So we were happy to see that the region that have decreased are not necessarily the most immunodominant. So essentially, to conclude this part, we look at our mutated epitopes in BA286, and what we realize is that once again, the majority of our original epitopes are conserved. And in terms of the mutated ones, in asking the question, what is the fraction that has a mutation that completely disrupt the binding or retain the binding, we realize that the majority still retain the capability to bind to the HLA. Now, there is a point that our analysis is not addressing, and I just want to mention that, which is TCR recognition. And so the fact that the peptide still is bound to the HLA and potentially present doesn't mean that the T cells, the specific TCRs, are still able to recognize it. So you can still have the potentiality that the TCR still recognize it or the TCR doesn't. An interesting alternative hypothesis is that you can have new TCR recognizing the mutated epitopes. And so that is a concept that has been explored in the past in literature, and we ourselves have some preliminary experimental data that support this finding. And so with that question in mind, we were wondering what happens if I simulate in prediction exposure with a Omicron sublineage, considering that the majority of the population has been exposed at this point. Am I going to see the same amount of difference across, or are the novel epitopes generated by an Omicron sublineage exposure kind of bridging the gap and so allow the T cells to keep up with the variants? And so this is actually the analysis that we perform. To be fair, in this case, we decided to perform a prediction based on the ancestral, the BA2, and the BA286 spike sequences. And then we ran this prediction using the most frequent class 1 and class 2 alleles to ensure population coverage. And so the selection of these alleles is something that IEDB has been previously curated really to allow the good coverage of the population. And so by doing that, now I'm summarizing the data. If we do the original comparison ancestral to BA286, the results that I'm showing you here are very similar to what I showed you before with experimental data. But now that we start doing the comparison between BA2 and BA286, you can see that the fraction of 100% conserved epitopes is much higher. And that is because, as we suppose, that the fact that the BA2 generate novel epitopes means that essentially those are much more conserved with BA286. So essentially, in conclusion, what we show in our results is that if you look overall to the spike and non-spike epitopes, once again, the non-spike regions are really the ones less affected by mutation. And this is why us and others are evaluating also strategy targeting non-spike region, potentially even for a pan-corona vaccine approach, considering that they are quite widely conserved across different coronaviruses. The second point is that should we be worried about novel sublineages from a T cell point of view? So our data by prediction and some of the data that are coming out experimentally suggest that indeed the novel variant epitopes can be generated following either breakthrough infection or bivalent vaccination. And so that can really bridge the gap between the original ancestral immunization we all experience or infection to the upcoming circulating variants that are going currently. So there is another story that I would like to share with you today, which is slightly related to this, but a step further. So I mentioned to you the generation of novel vaccine candidates. And so as we all know, in preclinical studies, one important component is to pass through animal models. And that is very important because it also provides us information about safety. But animal models are also used for immunogenicity. And so that raised the question to us, how much animal models can reflect the human T-cell immunogenicity, considering humans have such a variable HLA repertoire as compared to the mouse model. And so this is a study that has been done by my postdoc, Tertulliano Pereira Neto. And I would like to emphasize that when Tertulliano came in our lab, he was a pure experimentalist, never used IEDB, and after a few months, he was able to actually generate all this query pipeline, which was very impressive, but also suggests that you can do that too. And so what Tertulliano did was essentially to start with the epitope search. We focus on SARS-CoV-2 spike and nucleoprotein because we know the IEDB database has a lot of information about it. And then we use ImmunomeBrowser to really be able to map where the epitopes are located in the specific proteins. And then we perform this analysis by looking at the human data and the mice data, and we overlay them together to really ask, is there a good correlation or not between human and animal models? And so here I'm going to show you the results. On the left panel, we select all the human class 2 data as compared with H2B class 2 for mice, which are generally very frequently used in preclinical testing. And then we compare with specific HLA alleles that are available for transgenic mice, which is the other type of animal model that can be used to perform this type of experiments. And so what you can see, and on the right side, you have class 1. And so I'm going to essentially describe you a similar story. If you perform a correlation between the human data and the mice data, there is a significant correlation, but the correlation is very poor. The correlation can be improved if you start using transgenic mice, and so focusing on specific HLA, and it goes up to 0.59, at least in the context of DR1, and 0.4 in the context of A2 for class 1. One thing that I would like to drag the attention is that the correlation is in general throughout the entire protein. However, when we start diving more into specific immunodominant region, what we realize is that really the immunodominant region can or not be recognized by the different animal models. So here I'm going to show you a good example of that. So as you can see here in black, you have some peak of response for the human immunogenicity, which are completely unseen from the H2B mice, but they are, for instance, in A2 transgenic mice. But that works also the other way around. There is another good region for human HLA class 1 that is recognized, and H2B class 1 mice recognize it too, but that is not necessarily seen in the context of the A2 transgenic. A final point is, so far I've been talking about class 1 and class 2 like separate entities, but we were wondering, are those separately? Is it possible to find regions that are commonly recognized by class 1 and class 2? So our answer overall is yes, there is a good correlation or significant, although once again it really depends on the region, suggesting that when you perform some specific analysis, you're gonna be able to select specific region that could be common between CD4 and CD8 T cell responses. Is that specific just for SARS-CoV-2? Not really. We actually decide to expand this query to different viral systems, and the results overarching is always the same. What I'm plotting here are the correlation coefficient, and so here you see the data of the different viruses for the two transgenic possibility as compared to H2B mice, and the same for class 2. And as you can see, essentially, the H2B mice are the ones that perform the poorest. So in order to really understand these results, I would like to delve more into the implication of what we found. So we do have a correlation between human data and mice data, which suggests that mice H2B can be used for preclinical testing. It really depends what you're selecting. In general, overall, between H2B mice and HLA transgenic mice, we do see that HLA transgenic mice have a better correlation. So probably if we want to really have an estimate of the immunogenicity of the human population, the HLA transgenic mice are a better choice. Nevertheless, both for simple mice and HLA transgenic mice, it really depends what is the region that you are testing. And so in a way, if you were to design a vaccine, and probably you're trying to have a good population coverage of the human region, and you might select different regions that are important for that, the fact that those are not necessarily shown in mice doesn't necessarily mean that they're not immunogenic. And so that's something that is important to take into account. The second point is counterintuitively with what we thought before, it is actually possible to select regions that could be simultaneously targeted by CD4 and CD8 T cells. So that's an additional component that might be interesting, particularly when you do immunogen design. And then finally, this is not really a SARS-CoV-2 alone story, but what I show you so far is really applicable to different viral systems. And so with that one, I always like to say science is a teamwork, and I really would like to thank my team, my lab, and particularly Tertulliano that I show his study today. And of course, this was not possible without the collaboration of my mentor, Alex Fere, and John Simi. They've been terrific in most of the studies that I show you today. And thank you, IEDB, for existing. Otherwise, I couldn't even do this type of studies. So you are amazing. And my funding source. And with that, I will leave it to Alex, and I will be happy to answer any question in the Q&A. Thank you.